Hello and welcome back. In the previous video, we talked about the various partial pressures of both oxygen and carbon dioxide and how they're transported. What we want to do now is focus on the hemoglobin molecule itself and how it functions to transport these two gases in really amazing ways. So what is hemoglobin? Hemoglobin is a fascinating molecule and one of God's greatest creations. We think about it simply as a transporter, the ability to carry oxygen to tissues, and that's true. But what's amazing about hemoglobin is its ability to carry oxygen to the tissues that need oxygen the most. In other words, it can target its delivery to oxygen in a site-specific manner based on the environment. And we'll talk more about the details of that in just a minute. But let's look at the molecule itself. Hemoglobin is a tetramer, meaning it's made of 4 different subunits. 2 of these subunits are what we call alpha subunits. They're identical to each other. And then the other 2 are beta subunits, also identical to each other. Importantly, each one of these subunits contains a porphyrin ring, and this is where oxygen binds. We mentioned this ring earlier in talking about bilirubin recycling. I don't want to go into a whole lot of detail, but remember this ring is an iron-containing compound. This ring consists of 4 nitrogens, and it's with these nitrogens that iron will make coordinate covalent bonds. Iron sits right here in the middle. It's the iron that also physically links to the protein, to the globin structure itself, and then coming out from the top, this is what binds to oxygen. So the take-home message, each hemoglobin molecule contains 4 specific binding sites for oxygen. And of course, within a red blood cell, there are about 250 million hemoglobin molecules. And so that's a lot of oxygen carrying capacity for a given cell. Now, this structure, with 4 different subunits, endows hemoglobin with some very unique properties. The first one I'd like to list is cooperative binding. The best way to think about cooperative binding is to think about a 7th grade dance, where if we were to draw this out, we'd have one wall here, another wall over here. Of course, all the boys are hanging out here because this is where the table with the cookies are, right? And the girls are over on the other end, and they're hoping to be the first one to be picked. Of course, most 7th grade guys aren't interested in dancing, but along comes a chaperone and says, hey, why don't you go dance? So finally, one of these boys goes over and asks the girl to dance. Now we have a pair. Well, the competitive nature of boys is such that they don't want to feel stupid. And so now we see them move in mass to go ask the remaining girls to dance. This is the idea of cooperative binding. That first one is essential, and it makes it a lot easier for the rest to happen. Of course, the flip side is true as well. To let go of hemoglobin, we need to have a girl that decides she's done and she's ready to leave, and then everybody else will say, yeah, you know what, you're right. Dancing with these guys is kind of dumb, and so they all leave. But it takes that first one to initiate the effect. Now, what might this look like on a graph? If along the x-axis we're going to look at the partial pressure of oxygen, let's look along the y-axis at hemoglobin saturation. We'll do this in percent. Now we can actually break this up into quartiles simply because, remember, how many oxygens will one hemoglobin bind to? It has the capacity to bind to 4. So 100% is going to be 4 oxygens. Because of cooperative binding, if we're to trace this out, the hemoglobin saturation curve possesses a sigmoidal shape. To highlight the cooperative binding, as we trace starting at point 0, right, at 20, there's not a whole lot of action. But once that first one binds, then we see this rapid increase because the rest want to bind as well. So that's how cooperative binding contributes to the sigmoidal shape of this, or this dissociation curve. Of course, I want to highlight a problem. From our last video, we saw that, in general, the average PO2 of tissue, metabolically active tissue, was right around 40. And if we look at 40 on the graph, we recognize that it sits right up here, where 75% of the oxygen is still bound to hemoglobin. Hemoglobin's not letting go. In order to drop hemoglobin off, this curve shows me that I have to be down to like 20. And that's pushing really hypoxic tissue. That's painful. That's something that we're going to feel. This is where we see the magic of hemoglobin. We talked briefly a few minutes ago about how it can site-specific deliver oxygen to where it needs it the most. Well, let's take a second to think about what's going on in metabolically active tissues. Of course, the products of metabolism include carbon dioxide, so we have increased carbon dioxide. As part of this process, of course, carbon dioxide is in equilibrium with H+, so we have increased acidity, increased H+ concentrations. We also have increasing levels of a glycolytic intermediate, 2,3-BPG, bisphosphoglycerate. If you recall from glycolysis, the first redox step is the transition from glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, and this is just a cousin of 1,3-bisphosphoglycerate, a little isomer that actually serves this purpose of monitoring the amount of glycolysis occurring. And then the last thing, of course, we all know this from experience, increased metabolism means increased temperature. So these four things are associated with increased metabolic rates. What's fascinating is that hemoglobin has binding sites for all three of these. Of course, temperature is kind of ambiguous, but we understand how proteins alter with increasing or decreasing temperatures as well. So all four of these things will alter the structure of hemoglobin, one by temperature and the other three by physically binding to the hemoglobin molecule. And what that binding does is induce a conformational shape change that alters the saturation curve. What we can see is what's called a right shift. We shifted to the right. And we can see the effect that this has, because now at a partial pressure of 40 for oxygen, we've been able to let go of the majority of our hemoglobin. And so the difference from there to there, the ability to let off more oxygen from hemoglobin, this is what we call the Bohr effect. And where do we see it? Of course, we see it in the tissues, where we have this right shift caused by increased carbon dioxide production, increased H+ production, increased 2,3-BPG, and increased temperature. Now, you might ask, well, what about carbon dioxide? The Bohr effect is all about how carbon dioxide affects hemoglobin's affinity for oxygen. But can oxygen also affect hemoglobin's affinity for carbon dioxide? And the answer to that is yes. So let's set up the same scenario where we have a partial pressure of carbon dioxide this time instead of oxygen, but still looking at percent saturation on the y-axis. Now we're talking about carbon dioxide binding to hemoglobin, not oxygen here. In that same metabolically active tissue where we have high amounts of carbon dioxide, we see a saturation curve that looks like this. So remember, this is in tissue where we have high CO2 levels and, importantly, low oxygen levels. But what we see, if we were to look at this curve in the lung, where now we have low oxygen levels because that's what's causing this effect, we see a curve that looks like this. Now, a couple of things to note. First of all, there's no sigmoidal curve here. And that makes sense because CO2 does not bind cooperatively, as we discussed, with oxygen. It's just a linear curve. But you can see for any given difference in PCO2, very similar to the right shift that we saw with the hemoglobin dissociation curve for oxygen, we also see a right shift here. And this is called the Haldane effect. So if we were to summarize these two effects, if carbon dioxide is high, then hemoglobin's affinity for oxygen is low. However, with the Haldane effect, if oxygen is high, then binding to carbon dioxide is low. And these two concepts, the Bohr effect and the Haldane effect, working together, allow us to better understand the precise nature of how hemoglobin can site-specific deliver oxygen to the tissues that need it most, and then dump off carbon dioxide and reload with oxygen in the lungs.